Please feel free to use the chat box feature to send us questions. Questions will be answered to the best of our ability at the end of the webinar during the Q&A session. If we do not have time to get to your question, we will compile and consolidate your questions and we'll do our best to provide answers to you via the email that you provided for registration. My name is Kayla Cantrell. I'm a study director and toxicologist at the Institute for In Vitro Sciences. It is a pleasure to be your moderator for today. Thank you for joining us for the second of 2 webinars on the derivation of a point of departure from in vitro methods for use in skin sensitization risk assessment. We are very excited about this approach and its applicability to risk assessment as proposed by Dr. Andreas Natsch and his team. IIVS has been able to work with Dr. Natsch and offer this novel methodology as of last year, as well as offer all in vitro and in chemical methods discussed today. Our first webinar today will add additional context to part 1 of the webinar. We will begin with a presentation by Dr. Andreas Natsch from Givaudan about the calculation of a point of departure and how that fits into the 2 out of 3 defined approach, as well as hazard identification and classification. Following this presentation, Dr. Argel Islas Robles from the Institute for In Vitro Sciences and Dr. Andreas Natsch will walk us through some case studies involving examples on how to calculate the PEC3 for botanicals. At this time, I will introduce you all to Dr. Andreas Natsch. There we go. Andreas Natsch studied at the Swiss Federal Institute of Technology from 1988 to 1996 and was awarded his PhD in environmental microbiology. After postdoctoral studies in molecular biology at the Centro Nacional de Biotecnológica in Madrid in 1997, he moved to the research department of Givaudan in 1998. Here, he elucidated the biochemical mechanisms of body odor formation in order to design new deodorant ingredients. Since 2008, the research focus has shifted to the development of in vitro assays to study toxicological risk without the use of animal testing. This work focused first on the mechanism and kinetics of the reactivity of skin allergens with peptides, and then led to the development of the keratinescence assay to study allergens, which was globally adopted in 2015 by the OECD as the first biological animal-free assay to screen for skin allergens. In 2021, the 2 out of 3 testing approach and the kinetic DPRA developed together with BASF were adopted by the OECD. Parallel work of his lab led to 3 OECD test guidelines in the ecotoxicology field to replace acute fish toxicity test and bioaccumulation test. In his current position as Senior Research Fellow, he leads the in vitro molecular screening group at Givaudan to evaluate risk and bioactivity of novel ingredients early in the development pipeline. Dr. Natsch, thank you for joining us today. I'll turn the floor over to you.
Hello, everyone. I'm happy to be here again. For those who have already joined last week, to give you a second part of the webinar on sensitization risk assessment. So I hope you see the slides now in presentation mode.
Yep.
And we will start with the agenda of today's second part. I first will walk you briefly again to what we summarized last week, the model development and how it can directly be applied using a prediction spreadsheet. Then we will deep dive a bit more into data redundancy and the question whether we always need 3 tests to do a potency assessment, which directly leads us to the 2 out of 3 strategy, where we also sometimes have 2 and sometimes 3 tests according test guideline 497. And I will then show you how we can use the kinetic DPRA for the GHS subclasses assessment within 2 out of 3, and then directly to derive the point of departures. From that, we will go to some limitations, technical and applicability domain limitations, and have an outlook on further developments. And this will be on using human data as a prediction endpoint, but also the question of incorporating other tests as further input parameters. After Archil will then show you some case studies, I will then also go to some case studies on natural derived cosmetic ingredients to end this seminar. If you look back on what we did last week, so what I've shown you is that with all the key event-based test guidelines, we have next to hazard ID, continuous quantitative data, and that actually all this continuous quantitative data contribute to potency prediction. We use simple regression models. Other models would be possible, but we use regression models which can directly use continuous data to derive a continuous point of departure. So different to other projects which try to just predict classes, we look at continuous data for continuous predictions. And maybe the beauty of what we do here is that we don't need any additional testing. We directly take the data from the guideline tests, which have been done on hazard ID and potency classes, and we directly use the same data to do the point of departure. And finally, we have a simple public spreadsheet, and I will go quickly to it again, which makes application straightforward. And what I have also shown you, and we will deep dive more into that today, is with 3 tests, we already have data redundancy. And often 2 tests give us a very similar prediction as 3 tests. And I discussed already last week that this somehow could indicate that we are in a as good as it can get situation for a global model. So adding more tests will not further improve what we have. But at the same time, this also indicates us that we can work with partial evidence. So before I go into that in more detail, I quickly switch back to the spreadsheet just to remind you again. We have this spreadsheet where we just can enter the data which we have for 2 or 3 assays. So we can say we have a carotenosense. It was positive. It had a EC 1.5 of 20 micromolar and was cytotoxic at 100 micromolar. We have a kinetic DPRA with a log K of, let's say, minus 2.5. And as soon as we have 2 tests entered, we will have a first prediction here. That is equation 1, which integrates the kinetic DPRA and the carotenosense. If I would now say I don't have a carotenosense, but have an H-clad, which is positive with some data here, I will then have a prediction by the equation 4, which combines H-clad and kinetic DPRA. If I then would say I still have my carotenosense data, I will then have all the predictions combining carotenosense with kinetic DPRA, H-clad with kinetic DPRA, all 3 tests, or just carotenosense and H-clad together. And I have shown you that we have that also done for 2 case studies just to illustrate it. But why I just come back to that is just as a reminder, we have different models with partial evidence. That's equation 1, 4, 6, and 7. And we have the equation for the full evidence. And you will see equation 1, 4, 5 over and over again now with what is coming in the next few slides. So coming from that question on data redundancy, we come to the question of model choice. I have shown you we have different model combination, different models with different data combinations. And the question then is, which do I choose to make my prediction? And the first question here is, do I really need all the 3 tests? And that is the graphic here, which I showed you last week, that actually partial evidence combining two tests on the x-axis gives quite similar results as all three tests on the y-axis, which indicates, yes, we can work with partial evidence and we can work with two positive tests, as we sometimes have it in a 2 out of 3 assessment, where we stop after two positive tests and don't need the third test. Now, when we do that in 2 out of 3, just doing two tests, we have clearly shown that the sequence of testing will not affect the result. A yes, no answer from a 2 out of 3 is completely independent with which two tests we start. Now, of course, for the quantitative data, it is not so easy. We cannot just make this logical mind experiments. We really have to look at the data to prove that there is no big difference. So here we analyze what happens if we stop after two tests because we have a positive outcome based on two tests. Will it matter with which test we actually started? And I show you that here we have two positive tests, and we have started with the H-GLAT and calculated the point of departure by equation 4. And on the x-axis, we have two tests, and we started with keratinosense and used equation 1. And as you see here, overall, we have very similar results with a few outliers. But in general, it doesn't really matter with which test we have started if we stop with two tests. There is also the other case that we have three tests, and one possibility is that we didn't stop after two positive tests in a 2 out of 3. Maybe our strategy as a company or as a sponsor is that I want to have all the evidence and I always order all the three tests to be done. Then I might have three positive tests. And what is then the best model to choose? The obvious choice, of course, is I take equation 5, which integrates all the evidence. But more from a regulatory point of view, people might say, well, I want to be conservative and I take the lowest point of departure of the three models, which I have available with this partial evidence. With this full evidence, but also looking at those at the case where I have the lowest point of departure. And actually, that would give a very small difference. In most cases, when I take the full evidence of all three tests with equation 5, I end up very similar at a similar result as if I take the conservative choice to take the maximum PEC3, which is the minimal EC3. But I would say it is most appropriate to take all the evidence in case I have three positives. But the other case is a bit more tricky. If I have two tests which are positive and one is negative, what do I do then? One could, again, take equation 5, which takes into account that I do have negative evidence, because negative evidence is also evidence. Or I can take the model, which just looks at the two positive tests. Here is for those cases where I do have two positive. These are fewer cases in the database where I have two tests positive. And again, I compare the outcome if I take all the evidence on the x-axis and I take the evidence of the two positive tests on the y-axis. And as you see, the correlation is again very good between these two options. Here we do have a positive y-intercept, which indicates we are more conservative if we only take the positive evidence. That is somewhat logic, but it also indicates us that we are on the side of more over predictions when we ignore the negative evidence. But it also indicates the difference is not huge. So having done all these combinations and iterations, we can now go to the questions. What do we do if we combine everything together, the 2 out of 3 with the kinetic DPRA and this point of departure derivatizations? But at the same time, we also want to do the GHS subclassification, which actually are not the key subject of these webinars, but with the kinetic DP array, if we combine it with the 2 out of 3, it is quite noteworthy to highlight how we can combine that, because the 2 out of 3 is accepted to discriminate GHS 1A, 1B from GHS 2 from the non-sensitizers. And at the same time, the kinetic DP array is accepted to discriminate GHS 1A from 1B2 if it is in the applicability domain, which means if we combine these two tests in tandem, we have a fully validated approach to discriminate the 3 GHS classes, and we do not need any further validation for that. And how does this look in praxis? I show you a workflow where we start with keratinosens and the DP array, but you could equally well start with HClad and DP array in a similar workflow. So we do the first 2 tests, and if they are concordant and non-borderline, if we then have 2 negative results, we are done. It is a non-sensitizer. If you have 2 positive results, it is a sensitizer. And then we can check, is the chemical in the applicability domain of the kinetic DP array? And if that is the case, we can use the kinetic DP array directly to do the GHS subclassification. If it is not in the applicability domain of the kinetic DP array, or if we don't have 2 concordant results, we would conduct the 3rd test, which is currently the HClad, but there is work ongoing at the OECD to look whether other dendritic cell assays could be equally used at that point. And then again, we check, do we have 2 concordant results? If yes, if no, then we will need further data to make a conclusion. If we have 2 negative results now, it is a non-sensitizer. If we have 2 positives, and these 2 positives is the HClad and the keratinosens, we can directly know it's a 1B sensitizer, because chemicals which are negative in DP array but positive in the HClad. No, sorry, I have a wrong footnote here. I have to explain that why we have a 1B sensitizer when we have an HClad and the keratinosens which are positive, but a DP array which is negative, then we we will not have a positive kinetic DP array, but we would also be negative in the defined approach ITS. So as long as you don't have a positive DP array, you can have a sensitizer based on an HClad and the keratinosens, but it will always be a 1B sensitizer. But yeah, more details on that you find in the paper. If we have the HClad and the DP array positive, we are in the same situation we have been with the positive DP array and the positive keratinosens. If we are in the applicability domain of the kinetic DP array, then we can use the kinetic DP array for the subclassification. And if we are not, actually, then that's another case. You could then go to the ITS, which is also in test guideline 497. But with this workflow, you have the minimal testing needed to do a full hazard assessment and a full GHS subclassification. And that is normally all what regulators need. But now we go on and look at the point of departure. And as I said, the beauty of our approach is that you now need no additional data generation. You directly take the same data you have generated in your workflow. You just have to add the data to the calculations spreadsheet. So in case you have 2 positives, a keratinosens and a kinetic DP array, you have done it, used it for subclassification, and you directly use equation 1 to derive a point of departure. If you're in the other situation where you had to go on and do the HClad, and you arrive at a situation where you have the HClad and the keratinosens positive, you know it's a 1B sensitizer, and you can use equation 6, which combines H-clad and keratinosense, and you derive a point of departure. Or you're in the situation with H-clad and DPRA positive, you have the data from the kinetic DPRA, and equation 4 is used to derive the point of departure. But here in both cases, you will have all three data, and you could integrate also the negative evidence with equation 5, as I have shown you before. So that might look now as a lot of information quickly summarized here, but you can go through these workflows also as published in the paper. And basically, it's really a logical sequential flow of decisions you have to make. And when we go to the Q&A session, we might go back to that slide if anything is unclear in that workflow. So an important question which always comes up, what are the limitations of the approach we have here? When is the situation that we cannot apply it? And of course, that is very important. And one key limitation when we want to be accurate for quantitative prediction is if we are out of the applicability domain of the kinetic DPRA, then we might not see the full reactivity of a chemical, and which will lead to an underprediction. So one case which is clear, we see that for some aromatic amines, which have a lag phase for oxidation, and they might need first an oxidation step that we really see their activity. Another case are prohaptins, which require enzymatic activation. Normally, they are negative in the kinetic DPRA. And also, we know that selectively amine reactive chemicals might are underpredicted by this cysteine-based reactivity assay. Then there are chemicals which have a technical interference with the kinetic DPRA. They may or may not be assessed because this is the case for thiols, but it can be the case for fluorescent chemicals and chemicals which absorb in the excitation and emission wavelength of our probe. Now here, there is an alternative fluorescent probe described in the updated SOP. So in many cases, such interferences can be seen in a first assay, and then they can be remediated by using an alternative fluorescent probe. And there is also a third possibility. In principle, you can always do a classical DPRA and repeatedly inject your chemical into the HPLC over time, and then measure at exact times the depletion. And this can then, with the same mathematics we have in our spreadsheet, can directly be converted to a rate constant, which basically is equivalent to a rate constant in a kinetic DPRA. It's just much more time consuming to do that because you can only assess one chemical at a time, and you have to be very careful in programming your HPLC. So with that, we have summarized everything we have in the present. And I want to give you a short outlook on further possible developments. And one is, of course, what target data we use. Everything I have shown you so far is based on LNA data. So we have seen during all the OECD validation that it is very worthwhile also to look at human data, especially when we look at hazard assessment. And we also have some quantitative human data. So this is the first question. What is it if we go for human data? Actually, the data set of human data is quite small, and we would not recommend really to go and to train models only on human data because we could show that this data set is a bit biased. And I cannot show you that today, but you will see that in the upcoming publication. But there is an option that we combine human data and LNA data in a weight of evidence. And this has recently been shown by the IFRA IDEA team with Amaya as first author. Below you see the reference. What is called the reference chemical potency list is a list for 33 chemicals where we don't have an EC3 value, but something which is similar and is called the potency value. And it's also a continuous value. And this list only contains 33 chemicals, which is on the very low end to train such models with multiple input parameters. But I created recently a list with 139 chemicals with the same approach and did the analysis to see whether we end up at a similar model. And I did exactly the same thing. I did make a model for LNA, but I also made a model for this PV data. And this is now summarized here only very briefly, but it gives us two interesting learnings. First of all, that with human data, we have a different model where cytotoxicity suddenly loses much of its importance for predicting potency. But at the same time, you will see it gives us a very similar overall outcome. So first of all, I show you this here now only for equation 1, and I show it not as an equation, but just showing you the coefficients in the regression equation. And this is basically the equation 1, which I have shown you last time. And I did the same, train it again on this smaller chemical set here, again against LNA data, and the result is almost equivalent. So it indicates when I take this smaller data set, again, look at the LNA, the outcome is extremely robust, and we get the same model. But if I now take the same data set and take the potency value, which contain all the human evidence we can build in, then we see a difference for the EC 1.5 in carotenosense, which gets more weight and much less weight for cytotoxicity. So this tells us with this smaller set, we have a representative set with the same LNA model, but a slightly different model for the data with human evidence. But the interesting thing is now, if I compare these two models, the one trained on the LNA data and the other trained on the potency values, the overall picture looks very much the same for the moderate and strong sensitizers. I see that the very weak sensitizers are a bit different when I look at LNA or when I look at the weight of evidence data. But overall, this tells us we get very robust models also when we now take into account this human evidence. And our overall predictions remain very similar. This is just a brief overview of a bit longer paper, which will come out hopefully soon. And the other outlook is, now this is looking at the target data, animal versus human data, but the other question is, we see a lot of development at the OECD with new tests being integrated into these test guidelines. It will come quite crowded, the availability of different in vitro tests. And we already now, with the three first validated tests, have some illustrations of data redundancy. And I've shown you many of these graphs which show you that partial evidence or different combinations always lead us to very similar predictions. So I believe that mechanistic tests, additional tests, can improve local models. For example, in our lab, we have a special test where we evaluate aldehydes, or we have a test with S9 fraction in carotenosense for phenolic compounds. So there might be important other developments for such local specific tests for specific group of chemicals. But the key questions with all these redundancies, will additional tests improve these global models for all chemicals? And given that the keratinosense and age-clad are already quite redundant when it comes to potency, this puts a question mark, but at the same time, with the available database underlying our analysis, um, if somebody comes up with, I would say, at least 120 of the chemicals in this database of 188 chemicals, we can very quickly test whether the new test has orthogonal information or whether it just brings another test with a similar information content. That is now quite straightforward to test. And with that, I hand over to Archel for his case studies.
Thank you, Dr. Natch, for walking us through your publications in 2022, hazard identification using the PEC3s, and providing your expert insight surrounding analysis of test chemicals that require special consideration. We will continue to build on that knowledge with a case study presentation by both Dr. Argel Isles Robles, followed by Dr. Andreas Natch. Before we start our case studies, allow me to introduce you to Dr. Argel Isles Robles. Dr. Isles Robles has a bachelor's degree in biopharmaceutical chemistry from the University of Guanajuato, Mexico. In 2019, he obtained his PhD in pharmacology and toxicology from the University of Arizona. In the same year, he joined IIVS as a toxicologist and study director, overseeing assays for the assessment of ocular and dermal toxicology. In the last couple of years, he is focused on the skin sensitization approaches and currently is the program leader for skin sensitization at IIVS. Additionally, he works with government, industry, and other nonprofit organizations for the validation, implementation, and promotion of novel, non-animal strategies in toxicology. Dr. Isles Robles and Dr. Natch, thank you for teaching us today. I'll turn the floor over to you.
Thanks, Kayla, for the introduction. Just 1 second, let me share my presentation. And let me know if you can see the PowerPoint.
I can.
Okay, great. Thank you everybody for joining us for this second part of the webinar. Today I'm going to show a little bit more in detail some of the test cases using the approach that Andreas developed. And we have been using these regression models for about a year, in being in touch with Andreas and also with Frank Gerberick. So again, this will provide a little bit more details on how to make these calculations. And I'm going to stop sharing my camera just to save some internet. Okay, just as a reference, these are the two papers that came with the models that we are discussing in this webinar. And also for the test case that I'm going to show today, we use the nice spreadsheet that Andreas put together that is available as the supplemental materials on these references. So starting with the first test case, this is for 4-methylaminophenol sulfate or methyl. This is a chemical considered a prehapten, is used as a photographic developer, also in some hair dye formulations. This chemical is a proficiency compound for keratinosense, and it has a classification of GHS 1A, meaning that it's a strong sensitizer. In this table, I include a little bit of the physicochemical characteristic of the compound, and I just wanted to point out to the molecular weight and the vapor pressure, which are used for the calculations. Particularly, the vapor pressure is used in Pascal's units. Going ahead with the data for this chemical, we are looking right now at the KDPRA data. As a reminder, we use 6 different time points at 5 different concentrations that are plotted against the inverse logarithmic scale of the depletion. And based on this, the rate of the reaction is calculated as the log Kmax. In this case, we obtain this value, which means that we will be predicting a strong sensitizer according to the KDPRA prediction model. For the next in vitro test, we have the keratinosense. This is a typical graphical result of the assay, in where on this axis we have the fold induction, on the x-axis we have the concentration, and on the other side of the axis we have the percentage viability. In this case, this compound is positive on keratinosense. We can see how the induction crosses the 1.5 threshold. And for the parameters that we are going to use for the calculation, we have the EC 1.5, which is the concentrations that leads to crossing this threshold at 1.5 fold induction. And then the EC3 value, which is the same concept of concentration, but looking at the threefold induction value. We also are interested in the IC50, which is the concentration that lead to a decreasing viability of 50%. And moving on, in this little table, I just summarize these parameters that I just show. And we are looking at the freeze equation that has been discussed. This is using the data from keratinosense and KDPRA. And we see that we are using, of course, the reaction rate, the EC 1.5, the IC50, and the vapor pressure. I also wanted to point out that the final EC3 value is obtained through this equation, and please keep this in mind because I think that this is the only part of my presentation where I have this little equation. So overall, this is the calculated EC3 that we obtain using the equation number one. As you can see, the value is good correlated to a strong sensitizer. But what about if we have additional data? As Andreas has mentioned, it's typically a good idea to have all the evidence possible. In this case, I'm just highlighting some of the results for the H-clad for this particular chemical. This one was considered a positive. We obtained a CV75 corresponding to this value. As a reminder, the CV75 is that concentration that leads to a 75% viability. And then we also have the calculation of the EC 150 and the EC 200. The EC 150 is corresponding to the CD86 marker, and the EC 200 to the CD54 markers. And those are those concentrations that pretty much lead to a positive classification. In this case, we, for the equation, use the minimal induction threshold, which is the minimum value between the EC 150 and the EC 100, EC 200. So now our table that has all the parameters has grown a little bit, given all the data that is taken into account. I just add a little bit of headers to know which of the values or parameters we are looking at. So now I wanted to point out to the other equations that use the KDPRA in conjunction with the cell-based assays. Equation number 4 corresponds to the combination with H-clad. Equation number 5 corresponds to all the assays, KDPRA with the keratinosense and H-clad. For equation number 4, we are looking at the reaction rate, the minimal induction threshold for H-clad, the CV75 for H-clad, as well as the vapor pressure. And for equation number 5 is again the reaction rate from KDPRA, the minimal induction threshold for H-clad and CV75. And from keratinosense, we are using the EC 1.5 and the IC50, as well as the vapor pressure. And just to summarize the result is the table that again came from the spreadsheet that is available at the references. And the values that we are seeing here are also correlating to a strong sensitizer. They are a little bit different, again, given the different information that we are inputting for each of the tests. And then moving on, since we have all the data viable from all the assays, we have equation number 6 and equation number 7. This uses the data only from the in vitro cell assays, which are keratinosense and H-clad. And we have, of course, the minimal induction threshold and the CV75 from H-clad, and EC 1.5 and IC50 for keratinosense, and the vapor pressure. For these two equations, the only difference is the use of the data from keratinosense. Equation number 6, again, uses the EC 1.5, while equation number 7 uses the EC3. So looking at the results, this is what we are obtaining from the calculations. And on my next slide, I'm just summarizing the results from all the equations and all the possible regression models that we have. Here we can see again that all the values correlate with a strong sensitizer as expected. And I am showing here also the EC LLNA EC3 that has been reported previously. So as Andreas mentioned on his previous presentation, for selecting which value you want to use for risk assessment depends on the applicability domain of the assays. You can also be conservative and select the lower value or that equation that has all the evidence. Moving on with the next test case, this is for cinnamic alcohol. This is a prohapten that is found in cinnamon leaves, also is used in food and cosmetic industries as a fragrance and deodorant. This is also a proficiency chemical from keratinosense, and this compound is classified as GHS 1B, which is a moderate or weak sensitizer. Again, I'm including here the physicochemical characteristics. Just a reminder that we use the vapor pressure in Pascal units. And now that I am here, one good source of the vapor pressure is the EPA dashboard, which typically, if you have a CAS number, allows you to very straightforward obtain the vapor pressure. The vapor pressure can also be used from experimental data if available, but typically is getting from the databases. Here I'm showing the data from KDPRA. This is actually data that was obtained in our lab, and we are seeing that the result is non-reactive, and that pretty much means that we didn't see any reactivity above the threshold for positivity. That means that we will giving a classification of 1B or not categorized according to the KDPRA prediction model. And for the calculation on the spreadsheet, when we have a non-reactive, what we use is the default value, which in this case for KDPRA is corresponding to minus 3.5. And just wanted to mention that for all the parameters that are input for the EC3 calculation, there is default values in case that we have a negative or not calculated parameter. Moving on, this is the data from keratinosense. Again, in this test, it was predicted as a positive sensitizer, given that it crosses the 1.5 threshold and the viability is above 70%. For the specific values that we are going to use for the calculation, we have the EC 1.5, again, concentration that leads to crossing this threshold. We also are able to calculate the EC3 by extrapolating the data to this value. And similar thing for the IC50, using extrapolation, we can calculate how much is going to be the concentration that would lead to a decrease in 50% viability, which is around here. And finally, looking at the H-clad, this was considered a positive in the assay. I have here the CV75 and the EC150 and EC200. All these two values are input in the spreadsheet. However, only the minimal induction threshold is used for the calculation. And just as a reminder, this is the minimal value between the EC150 and the EC200. So looking at the table that has all the parameters, we have, again, physicochemical properties and the parameters for the three in vitro assays. So just very quick, in this time, I'm not going to show the equations, but you can look at the previous slides. We have here all the values calculated from each of them and also the reported EC3 value. So again, this correlate again with moderate or 1B, GHS category 1B. So this was the last case that I wanted to show. If you have any additional questions, feel free to ask in the chat box. The questions are going to be responded at the end. And then also you can email me if you have further questions. Thank you.
Andreas, we can see the PowerPoint, but we cannot hear you at this time.
Now you can hear me. Sorry.
No problem.
Yeah. Okay. Perfect. So there is a big question always asked very often is what about testing naturally derived cosmetic ingredients, which are often enriched single constituents or complex mixtures. And in principle, for age-clad and keratinosense, we have a gravimetric prediction model validated, so we can use something where we don't have an exact molecular weight, and then we make a solution basically based on weight. But this is not the case for kinetic DPRA and DPRA, where this has not been validated. So for a really validated prediction, we cannot use these combinations so far. But often when it comes to making a prediction for a complex cosmetic ingredient, it's not really for registration of this ingredient. Often these ingredients are below the threshold, volume thresholds where you need this full assessment for regulators. But very importantly, we need to make a risk assessment for those cosmetic ingredients to really find safe use levels. And that's why, in absence of any other validated tests, it is still very, very valuable to use the tests we have and do an assessment, keeping in mind that also all the animal tests were sometimes tested on few complex ingredients, but they were not validated specifically for complex mixtures. And when I talk about complex mixtures, I don't mean finished products. I think all these in vitro assays were never validated or meant to be used for finished products. But complex cosmetic ingredients can be any plant extract, which is then added to a cosmetic formulation. So I show you test item one. I cannot really say what it is here because it is something which we developed in-house here, but I show you just the data. So this was very clearly positive both in keratinosense and it was reactive in the kinetic DPRA. You see here the kinetic DPRA results at different time points, and we see a reactivity increasing from the 10 minutes to 24-hour time point. And we see that this chemical only gets significantly positive at the 24-hour time point, which gives a rate constant of minus 2.55. And at the same time, we have a positive keratinosense, which in the spreadsheet can be directly added as a ppm value. If we don't have it in the micromolar here, we add it directly in the ppm here, and it is transformed automatically here, assuming a molecular weight of 200, which is the default molecular weight for complex mixtures. When we combine that, we find an EC3 value of 3.6%. And why I show you this specific case study is because here we could find literature data. These were literature data from the same plant, from an extract from the same plant. Of course, we never know whether it's really the same test item because it was an extract which is not fully characterized in the publication, and we have our own extract, but it's a similar quality extracted from the same plant. And in that literature citation, we found that it reacted at 5% both in the LNA and in the mouse ear swelling test. And this is pretty much in line with this moderate sensitization, which we've seen here based on the keratinosense and the KDPRA data. And I show you 1 second. Oh, sorry, that was the wrong button. And I show you a second case study. This case was strongly positive, but just below the threshold in the kinetic DPRA. We actually found 8.8% Swiss depletion in the kinetic DPRA at 24 hours, and also 8.9 cis depletion in the classical DPRA. So it is just below the threshold of 13%, very weakly reactive. This is the full depletion matrix. You see there is very little reactivity. It starts at 24 hours, but below the threshold. So we have the default negative threshold, which for no depletion above 13%. And then for the keratinosense here, we have a very strong result already at 0.3 ppm. It was positive with also a low IC50. And based on that, we again find a moderate sensitizer with an EC3 of 2.5%. Also, I show you this second case again because we found a publication where we have something where we can make a comparison. And in that case, we found a study with a guinea pig maximization test where this chemical was tested at 5%, and 60% of the animals were sensitized to the same active principle. Of course, again, we don't know the exact purity in the publication compared to our purity, but it shows that a very similar preparation gave a result which was quite aligned with our in vitro estimations. These two cases just illustrate, yes, we can test mixtures or enriched ingredients from plants which are meant to be used in cosmetics. We can, in those two cases where we know historical sensitization results, we find similar results. So technically, this is possible, and we can at least use it for a risk assessment. Then we, of course, need very clear safety margins when we find such results of moderate sensitizers. After these case studies and Orchil's case studies, I come to a quick summary of what we discussed today, and then we have plenty of time for questions. So what I showed you is based on the data redundancy, 2 positive tests can be enough for a point of departure, and the sequence of testing is not critical, which means that additional tests will often not further improve the correlation for a global model possibly. And we should probably in the future design new tests for specific chemicals, rather more global tests. But I put that with a question mark, and evaluation in the coming years of the new tests will tell us more about this. At the same time, I showed you that we can incorporate the kinetic DPRA in the 2 out of 3 assessment, and with that we can make a full assessment. We have hazard, yes, no, is it a sensitizer? We can have GHS subclasses, is it a weak or strong sensitizers, fulfilling all the regulatory needs, and then directly use the data for the point of departure. And this is all determined in one workflow without additional separate testing. And I showed briefly, gave you a teaser for what I've done very recently. What happens if we add human data to the same assessment? And the good news is that overall we have a very stable model with human data that will also be added to the prediction spreadsheet. So there will be an update of this prediction spreadsheet where you also can predict the weight of evidence versus human data. It gives very similar results. That's the good news. The more scientific interesting news is that we have a different weight for cytotoxicity and cell activation, which gives us indication of some mechanistic differences between the LNA and the human data. And finally, the case studies show that we can apply these tests also to more complex ingredients. Of course, the less defined our test item is, the more uncertainty we do have in our assessment. For the acknowledgements, it's the same as last week. I worked with Frank on the publications and the scientific discussion about the whole approach. With Roger and Tina in my lab for a lot of the database work together with BASF on validation and database, and especially with Suzanne in the long and epic fight for the validation of the 2 out of 3 and there was a lot of data curation and compilation done by the OECD expert group. Happy to take questions.
Thank you both for walking us through these case studies. Undoubtedly, we must understand the applicability challenges of this method as well as the method itself. As Andreas and Arhel said, at this time we will be taking questions. Remember, you can use the chat box to type your questions, and for questions not answered, we will do our best to answer and compile the questions and share the questions and answers to all attendees in the email you use for registration. But considering that we have about a half hour, I think we'll get through them. So our first question came from Pierre. You mentioned limited solubility is an issue for KDPRA. Can you quantify a threshold?
I would say the brief answer is no. um, in general, it's often mentioned that yes, if your chemical is not sufficiently soluble, you can't test it. Now, for the DPRA, there was a lot of comparison made between the DPRA and the ADRA, because the ADRA is done at much more dilution, and the developers of the ADRA claimed that this makes it more predictive. But when they then compared DPRA data for those chemicals where visually you see that they are not dissolved in the DPRA, you see turbidity, and they compared the predictivity for the ADRA where you have a better solubility because you just dilute more, the predictivity was actually the same. Because when you have an excess of your chemical and it's not soluble, you still have a saturated solution for reaction. And then your peptide, as it starts to react, there will always new chemical be able to dissolve. But of course, this is not you don't have the same amount of chemical available for reaction at a given time as if you have the same chemical completely solubilized. So what it really means for the quantitative value from the kinetic DPRA, it's quite difficult to quantify that. What we see is that we have quite poorly soluble chemicals which still give adequate reactivity, especially when they are strong sensitizers, and we measure then the key time point can be the 10 or 30 minutes time point, and they may still be in a stable dispersion, allowing for good reactivity. But I think we cannot make an absolute quantification. And at a certain level, the solubility will be too low for an adequate result.
Thank you, Andreas. And I just wanted to add a little bit more on perspective. We actually have had this similar case a few times while we are trying to do these calculations. Of course, the KDPRA uses set concentrations. For example, the top stocks are prepared at 20 millimolar. There is one option to use lower concentrations until you see that the sample is very diluted, because as Andreas mentioned, you want the sample to be diluted so you can quantify correctly. As I say, you can use lower concentrations. However, the classification according to the validated tests are not going to be able to be used for regulatory purposes, but maybe if you still have a good solution, you can use that, to estimate the reaction rate.
Thank you both. Our second question comes from Cindy. If you use the equations derived by incorporating human data, do you still do an uncertainty assessment and apply a factor of 2 or 3?
Yes, I would still add an uncertainty factor, because it is not in vitro versus animal versus human. It is directly in vitro versus an assessment integrating human data. But because we see quite similar predictions, I would still keep the same safety factors.
Our third question comes from Stefan. Is it generally possible to use the in vitro test to test products containing active ingredients with a pharmacological effect? If so, are there pharmacological modes of action that are known to cause issues with some or all of the test?
In this case, of course, it's possible to use the test for different types of products, but the most important thing is always try to be aware of the limitation of the assays. It's always important to be on the applicability domain of the assay, but of course there can be some type of material that causes interactions. For example, in DPRA, the DMSO is able to dimerize the peptide, and that's one reason why it's not a good solvent. So can imagine how some pharmacological compound will be able to, for example, dimerize the peptide or perhaps oxidize the peptide in where the results will be questionable. So it is possible to use in vitro tests to assess this type of material, but of course we need to take into account the applicability domain and how each of the assays will perform in these cases.
And overall, quite little pharmacological items have been tested in these assays because in the pharma world, using animal tests is still more routine than in the cosmetic world. But lately there has been, for example, a lot of work looking at pesticides. So the pesticide sector has looked at these tests. And for example, the carotenosense has worked quite well for pesticides. Which have very different modes of actions and not pharmacological activities, but very specific toxicological mechanisms. Yeah, and there are some pharmacological chemicals which have been tested, but there's quite little experience.
Thank you for that perspective. Amelia says, very good case studies. Thank you to both presenters. Besides the OECD guidelines that provide info on applicability domains for each of the available in vitro methods, is there a library of compounds publicly available to share which compounds may interfere with what method?
To my knowledge, I don't think that there is any specific library in where you can find information. However, you can find other resources that give you a little bit more information on the chemical, and then you will try to compare if it is in the applicability domain. One of them is the OECD toolbox. For example, in that case, you can have information if you have prehaptens, prohaptens, if you have any material that might be oxidizer. So again, to my knowledge, I don't believe that there is any library that specifically tell you which assays you will have an interference depending on the chemical. But again, there is other resources where you can look at the chemistry of the compounds.
Actually, for the kinetic DPRA, it was the first time that we generated a tabular a table where we said what is the reason, what type of chemicals are outside of the technical or prediction applicability domain, and we gave examples. So this table was I think that's the first guideline which has this table, at least in the sensitization field, but not like really an inventory of chemicals. That's not done so far.
Cindy asked, in Andreas's second case study, there were non-concordant results between the KDPRA and the carotenosense. Why was an HCLAT not conducted?
Yeah, that's a good question. Actually, this was really a chemical which we were developing, or a mixture which we were developing. And in that case, we only do the in-house assays which we have, which is DPRA, KDPRA, and carotenosense. And only when it goes, when we need further assessments, we would go for an HCLAT. But as you could see in that case, we already came at a quite good assessment with these two assays. And it was not meant for a regulatory submission.
Adrian asked, speaking as a regulator, I am most interested in the classification result, but can you help me to understand the benefit of a POD or say more about how it's used and practiced by industry? I work mainly in agrochemicals, so particularly interested in this area.
Yeah, I mean, in praxis, in the fragrance industry, we developed, and that was led by the Research Institute for Fragrance Materials. In the fragrance industry, we use the QRA and now the QRA2, which is the quantitative risk assessment. And there we start with what we call a NESIL, a no expected sensitization induction level, which comes from an LNA and then combined with the human no effect level in the HRIPT. And this no effect level is what we use as a starting point to then define what is a safe use in a particular product. And from our point of departure, which is a mimic for the LNA EC3, by applying a safety factor, normally I take a safety factor of 3 by default, we would have a predicted NESIL, which we then can also apply in this QRA. So this is really to define safe use levels in a product. And very similarly, that can be done in the pesticide industry. In the pesticide industry or in the pesticide regulation, there is not yet something defined like the QRA, which we use in fragrance and cosmetics. I was on a workshop in Basel early this year where people convened regulators and experts, and it was discussed how this could be applied actually to the pesticides. But the exact safety factors for pesticide workers, they need still discussion, but that's where this point of departure would feed in.
And I also wanted to add, working from the perspective of clients, I think that these different questions, like one is the hazard assessment, in where they are interested into knowing the classifications for labeling or other type of regulatory purposes, and there is a question about potency, like as Andreas mentioned, to assess those levels that is safe for having a sensitizer in the products, but pretty much are separate questions that typically our clients have. One is hazard assessment, and the other one is potency, and the EC3 allow you to have that potency data from the in vitro data.
Cindy asked, is there any work being done to use other assays besides keratinescence and HCLAT? For example, LuSense data in place of keratinescence data or USense data in place of HCLAT data?
Actually, I haven't done that. I would say for the LuSense, we compared it once, it gives almost the same results. There's just one problem with the LuSense, that it normally measures a more narrow window of concentrations, and sometimes you don't have an EC 1.5 value if you don't hit the right window. That could be a problem in some studies, but in general, it will give a very similar results. These data correlate very well to each other. For the USense, it would be easy to do. I just would need the Excel sheet with the data and the CAS numbers, and I could of course source that from the literature, and that would be quite easy to do. But I haven't done that so far.
And also, Andreas, correct me if I am wrong, but I believe that you need a big database on the results. And I'm assuming that there is a lot of knowns for HCLAT and keratinescence, and probably for the other assays, there is not that many, non-results or public results.
For the U937, there would be enough results. I have seen the publications. I just haven't sourced them. But there would be enough data. The key question would be now for the GARD potency, the GARD dose response potency, which is sort of promoted as a standalone potency assay. There I haven't seen a sufficiently big database to do this test, but this will, I think, very soon will be available, and then we can compare that also.
At this time, we have no further questions in the chat, but I did have a question for you, and that should give anybody who had a remaining question some time to put that in the chat. You mentioned that you can use the negative 3.5 if no Kmax is calculated in KDPRA. How is the negative 3.5 calculated? Where does that number come from?
That is actually if you're at the threshold, if you in your highest concentration after 24 hours, you have, I think I took 12.5 or something, just below this 13%. And if you add that to the formula, then it gives you a 3.5. And so we took the minimal reactivity which you would see as a threshold, which is a conservative choice. You could also take half of that, of course, as a less conservative choice.
Thank you. It doesn't seem that we have any additional questions at this time, so thank you both for your time and presentations and helping us understand this method.
Thank you all for your interest, and I think the IVS will share the presentations for those who haven't understood everything already.
Yeah, thank you for attending. And I will be sharing the slides as well as the recording in a couple days, probably.
Okay, see you all.
Thank you.
Goodbye, everyone.
Goodbye.